Not,
Is everything good?
not yet. Not yet. Try to demonstrate once again. Share screen on your right, and then share the main generic PNP monitor. That's what I hit mostly. And I click the toggle with sound. So use the sound from my computer. Yes, I can see it now.
Okay. Now
Yes, Vitruvian men. Yes.
Yeah. Okay, so today's talk is epigenetics, and I'm going to talk about what it is, how it works, and what it means to you as an individual. So first, what is it? There are 4 adaptive mechanisms that all organisms that are eukaryotes in nature, and all colonies of organisms and all species have to use for adaptation so they can survive. There are homeostasis, neuroplasticity, epigenetics, and natural selection. So homeostasis is immediate reactions to various stimuli. Like if you get cold, you get goosebumps, and that helps retain heat, and the hairs on your arm will stand up, and that also helps you retain heat. If you get into a competitive situation, you have to generate adrenaline. You have to do it very quickly so you can be more competitive. If you're being threatened in some way, you have to generate cortisone so you can do the fight or flight business. If you sense hormones, you have to react to those immediately. And this is survival in the short term, the very short term, and it's on the individual level. So it's all about you. You're in a situation, you have to immediately respond to it. So you have to change immediately and rapidly. But it's very short duration. It only lasts seconds or minutes or at most hours, and the effects are temporary. When you're no longer cold, you go back to the way you used to be. When you're no longer in competition, your heart slows down, and so on. For longer term issues, though, neuroplasticity is the basic premise that keeps us going. And this has to do with brain development and the way we use our brains. So when you're born, your neurons are connected to every neuron that's nearby. And as you experience stimulus, your dendrites actually break connections. This is called dendrite pruning. And it's the way your brain organizes itself as you mature from an infant into the first couple of years of your life, where your brain is organizing itself rapidly. Also in your brain there are neurons called mirror neurons, and these are used to enable you to learn by watching or imagining yourself doing things. So if you see somebody playing tennis, these neurons fire in your brain, and your brain is actually practicing doing what you're watching. So you can actually learn a lot about how to play tennis just by watching somebody else do it. The next level is muscle memory. This comes from practice. So when you're learning how to walk, muscle memory is retaining this practicing of you walking and this improvement in how you walk. And as you're doing this, a program is being written in your brain that's downloaded to your cerebellum. And then later on, when you know how to walk, or you know how to ride a bicycle, or you know how to play the piano, what you've got is these programs that are programmed into your cerebellum. And when you're given a certain stimulus, your cerebellum runs that program. So if for example, you have to run somewhere, you don't have to think about running left foot, right foot, left foot, right foot. You just trigger the cerebellum to run the running program, and it becomes a reflex action. You don't think about it at all. So I play the guitar, for example, and I can play the guitar and talk to somebody at the same time because I'm running this program that says, here's how I play this song on the guitar. And that leaves the rest of my brain free to talk. And then the final level is consciousness. All of your stimulus inputs come into your brain. They're distributed throughout your brain. and your brain processes visual input in one place and auditory input in another place, and smell and other sensory input, and your brain has to bring it all together, and that takes place in the precuneus, and that's a part of your brain that's kind of at the top back of your head, the pointy part of your head, in fact. And then that goes to the prefrontal cortex, and that's where you see what happened, reacted to it, and that's where you say, well, I'll never do that again, or that worked, or that tasted good, and that also feeds into survivability, right? So all of that is for your survival at the individual level, and also the survival of your relatives, who you want to protect and help and so on, and also whatever tribe you're in, it helps them survive as well. And these changes are lifelong. Once you learn how to ride a bicycle, even if you stop riding a bicycle for years, you can get back on a bike and you still know how to do it, because that program is still there in your cerebellum. So the duration of the effects of neuroplasticity lasts for months or years, and the effects are your entire life. Natural selection, you know what that is. This is about individual selection, and we're again talking about survival so that you can procreate, and that's how natural selection feeds into further generations. Sexual selection plays a big role in that, because you have to select a mate that's going to aid in the viability of current and future generations. So selecting a mate becomes a very important role, has a very important role in that. Also, that feeds into kin selection. And this is, who are your friends and who are your enemies? And how do you treat friends, people like you? And how do you treat people who are strangers or not like you? And then group selection. And, how do you protect your friends? How do you protect your family? And how do you compete with other family or other groups? And this is, again, survival of the species. The viability of current and future generations is the key phrase there. The changes are what we call evolutionary, although that's largely misunderstood. It's really about how do you create, how do you procreate, and how do you procreate in such a way that current and future generations have higher survivability. The duration is years or thousands of years or eons, and the effects are as permanent as they can possibly be, definitely multi-generational. But there's a piece missing. So homeostasis covers seconds, minutes, and hours. Neuroplasticity covers days, months, and years. Natural selection covers millennia, millions of years even. But epigenetics fills the gap of decades, generations, and maybe even centuries. So epigenetics is the part of our adaptive mechanisms that enables us to build an organism, like a human or a dog or a butterfly, and all of its parts and basic settings, and I'll explain that more later, in response to the current environment. So, for example, if you live in an environment where the temperature is on average 70 degrees, you will behave in a particular way. If food is plentiful, you'll behave in a particular way. But if food becomes scarce, how does your body adapt to that scarcity right now? And how does it prepare your next generation and the generation after that for potential scarcity? Then there's episodic things you have to deal with, like you go into puberty. Well, your body changes when you go into puberty. So how does that happen? How does it know when to go into puberty? How does it know when puberty is over? And what changes happen then? And how does that get triggered? A bear has to fatten up for the winter and then go into hibernation. Well, how does the bear know? And how does hibernation get kicked off? And so on. And then there's the whole issue of sexual selection, because sexual selection in humans in particular changes depending on whether a female is an estrus or not, whether a male is high on testosterone or weak on testosterone. And all of that's going to impact further reproduction and the viability of current and future generations. And all of that stuff is being triggered all the time. So what triggers it? And how does that work? Then this is survival at the seasonal, the episodic, and generational levels. And the changes then tend to be seasonal or episodic or generational. The duration, same, and the effects, the same. Now, back in 1944, we discovered as humans that genes are made of DNA. And we thought at that time that DNA did everything. And DNA is a sequence of genomes, and it's a fixed thing. So if DNA controls everything and it's fixed, then you are the victim, more or less, of your DNA. If you were born to be tall, you would be tall. If you were born to be short, you would be short. Curly hair, blue eyes, all of that is fixed by your DNA. And that's all they knew. All the scientists knew about how we were made was based on the discovery of DNA. Okay, there it is. So I'm going to show you a little video about how DNA works. And this is extremely interesting. So Avi, let me know if this video does not work, but otherwise just let it play.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Okay, so a gene is read by RNA polymerase, and it turns into a strand called messenger RNA, mRNA. That's read by a ribosome 3 letters at a time, and every 3 letters produces an amino acid. And one gene then produces a chain of amino acids, which folds up into a protein. And this is referred to as the central dogma of molecular biology. We know this is how it works. Now, proteins and non-coding RNAs do everything. And I'll talk about that more later. But protein holds oxygen in your red blood cells. Hemoglobin is a protein. Salivary amylase is a protein that breaks down sugars in your mouth. Insulin is a protein that regulates metabolism of sugars, fats, and other proteins. Actin and myosin are proteins that contract and relax your muscles. So even moving requires proteins to be produced. NMDA receptors in the brain control learning and memory. These are proteins. Proteins and non-coding RNAs do everything, right? But how does the RNA polymerase know when to go read a particular gene? How does it know when to do that? And how does it know which gene to go read? And that's the big mystery. How does it know how much of a protein is to be produced? Because if you're going to produce a particular protein that performs a particular function, you don't want to produce too little of it, and you don't want to produce too much of it, because those could be problems. Also, some of these proteins you want to produce for a period of time and then stop. So how do you know when to stop? Also, when you look at DNA, you find that there are only about 21,000 protein coding genes, the genes that make proteins. And that's the same for most animals. Worms, flies, fish, all have about 21,000 protein coding genes. So there are 3 billion base pairs of coding genes in your DNA. So only 20% of the genome is used for coding proteins. That means 80% is doing something else. Well, 20 to 40% of that is for gene regulatory sequence. And that means that there's still some left over. We don't know what it does yet. But 90% of genes is transcribed. So all of that stuff, all of that information is necessary. Well, the genes that code for proteins, you can think of in computer terms as the hardware interface, right? When you start up a computer, it knows how to boot. You don't have to program it in. You don't have to load software. It's built in. And that layer of information inside your computer is what the software that you run talks to, that drives what the hardware does. So that's what 20% of your genome is doing. And that interface to the hardware is those coding proteins. Because remember, they're the things that cause your muscles to flex and your eyes to see and your ears to hear. The 20 to 40% is kind of the system software. When a gene is being read, there might be an error in the gene sequence that has to be corrected, or the gene might have to be reorganized a little bit in the RMA stage before it can be used. And that's what that stuff is for. So there's 30 to 60% left over. And they used to think, scientists used to think, and they used to call this junk DNA. They didn't know what it was for. They said this must be left over from some evolutionary process that we don't do anymore, and it's just garbage. Now, before cells differentiate, they have the potential to be any type of cell. At this undifferentiated stage, they are embryonic stem cells, right? They could turn into anything. They could turn into brain cells or blood cells or muscle cells or bone cells or liver cells. So how does a cell know which genes to express that make it a muscle cell or a brain cell? All cells have the same DNA, so they all have the potential to be any kind of a cell. So something must trigger that metamorphosis into the appropriate thing. And cells go through stages. When an egg is fertilized, it starts out as a single cell. Then it starts to divide. And it divides over and over and over, multiplying by factors of 2. And eventually it turns into something called a morolla, which is Latin for mulberry, because that's what it looks like. So initially, they're totipotent. They can become any type of cell: skin cell, blood cell, neuron, pancreatic cell. So let's say there are protein coding switches inside the cell, and they start out in a standby position. So the cell can be anything, but the switches all start out in a neutral position. Then they become pluripotent, when they can become one of 4 types of cells: endodermal, ectodermal, mesodermal. There's 3 types, sorry. Endodermal could be thyroid, lung, pancreatic, liver, gut. Ectodermal, skin, neurons, or brain. And mesodermal, skeletal, blood, or circulatory. So when they're going to become one of these 3 types, there's a switch that has to get set that says this is a mesodermal cell. And it goes through stages, right? As the cell divides and divides, it goes through stages where first it determines, is it going to be a placental cell, and it's going to be on the outside of the egg, and it's going to be the part of the cell that attaches to the mother, or is it going to become a fetal cell? And as a fetal cell, it's going to start to become the creature itself that's going to be born later on. And if it's a fetal cell, well, is it going to become an endoderm cell or an epiblast cell, a pluripotent cell? And you can see in the pictures that these things happen at different points in time related to how many cells there actually are as it's going through its divisions. And over time, different sections of the organism start to develop. The central nervous system develops in the first 3 weeks of life, where teeth don't start until the 6th week of life. So they get triggered at different points during development. When they go into the pluripotent stage, right, we're going to say some set of cells or some set of triggers, switches, get switched on. So if it's going to become a mesodermal cell, the switch for mesodermal gets turned on, and the switch for endodermal or ectodermal, they have to get turned off. Then we go into the multipotent stage. In the multipotent stage, if it's a mesodermal cell, it could become skeletal, muscle, cartilage, bone, or fat cells. If it's going to be an endothelial progenitor, it could become an artery or a vein. If it's a cardiac progenitor, it's going to become heart cells later. If it's a hematopoietic cell, it's going to become a white blood cell or a red blood cell or a platelet. So let's suppose then it's going to become an endothelial progenitor. So the switch for that gets turned on, and the switch for mesenchymal, cardiac, or hematopoietic, which is hard to say, they all have to get turned off. Now it can become an oligopotent cell, which means it's going to become one of the limited types of cells within that system. So in this case, it's going to be either an artery or a vein. So let's say it's going to be an artery, so now we have to turn on the switch or switches for artery and turn the switch for vein off. And then it becomes a unipotent cell. Now when it's a unipotent cell, it's fully committed and it's self-replicating. So if it's an artery cell and it divides, what it divides into is 2 artery cells with all of the switches set to be an artery cell. As long as this works correctly, every time it divides, it's going to create 2 artery cells and nothing else. So how do the switches get turned on and off? And how do the dials get adjusted for intensity? And when do you know when it's time, or for how long a switch stays on and when it gets switched off? And if it gets switched off or on at a particular time, how does it know when the time is? And then when it divides, how do these switches get copied in each cell so they're identical? And so every time the cells divide, the switches are always identically set so that we don't get a mistake and an artery cell turns into something else. But there are no switches. There are no dials. There are no timers. There are no sensors, and there are no operators. Or are there? There's only DNA. And DNA creates genes. And genes are read into proteins and non-coding RNA. So every cell type is defined by the genes that are expressed. And remember, every cell has exactly the same set of genes. So that means that in an erythrocyte, a red blood cell, maybe the gene we call gene A is turned off. Maybe gene B is turned on and it's expressed in a large extent. And gene C is turned on, but it's not expressed quite so much, and we end up with a red blood cell. For a neuron, it's different. More of gene A, a little of gene B, and some gene C, and so on. If things go wrong, it can be really bad. Here's an example of an insect where legs are growing where legs should not be. Okay, this is pretty disastrous, but these things do happen. So when things go wrong, they go very, very wrong. Well, this is a picture of a chromosome. You saw that before. Inside every cell of your body, there are 2 meters of DNA. So they can't just exist as long strands. There's no room for them in that state. So they have to be wound up really, really tight. So there's a structure for each chromosome so all the DNA can fit inside the cell. And if we zoom in on that, let's see. If we zoom in on that, here's a piece of the chromosome. And if we look at it really closely, what we see is the DNA in there makes loops. And it's all looped up inside the chromosome. And if we look at one of these loops, or a section of these loops, what we see is there's a chromosome scaffold, and at the bottom of each loop, it's attached to that scaffold. And this is used to separate the loops. So you can do something that impacts one loop, but does not impact the loop next to it. And then if we zoom in on these loops, what we see is they're made up of chromatin, which is, if we expand it a little bit, we see it's DNA strands that are wound around a bead, and then it goes on and it's wound around another bead, and then it goes out and it's wound around another bead. But the space in between those beads, that's the DNA that's going to get turned into proteins later on. So these beads are made up of proteins called histones. And this group of histones over here, that group of histones, that's called a nucleosome. And there's 30 million of these in every cell. And the function the nucleosome provides is key to epigenomics. The DNA that's going to be described is the DNA that's between the histones. This is what it looks like. This is a computer model. Here's the DNA wrapped around the 4 proteins that make up the histones, and you notice they're in pairs. Here's one that's indicated as yellow, and there's its pair, yellow, over here. Here's the green one, here's the other green one. So there's actually 8 of these proteins, but there are only 4 types. Each histone has a tail coming off of it. And these tails are the things that activate and regulate the expressions of genes and also deactivate the expressions of genes. The histone tails are packaged in all eukaryotic DNA, from yeasts to humans. This is the way every living thing that evolved from eukaryotic cells works. So it's a very, very old mechanism. Now, these histone tails have spots on them. This was discovered in 1964, by the way. So pretty recent. These histone tails have places on them where a tag can be attached. And this tag is attached by an enzyme that plants the tag onto the tail. And that's called a writing enzyme. And an enzyme can come along and remove it, and that's called an erasing enzyme. So these enzymes come along, they add tags, and they can come along and delete tags. So they tune what happens inside the DNA by using these tags to create proteins temporarily for the processing of something like alcohol, maybe, or episodically. So when a baby is nursing its mother, at some point it becomes lactose intolerant. And that's why babies wean themselves away from breastfeeding. So at some point in its development, a tag has to come along and make that baby lactose intolerant by producing a protein that causes that effect. And some of these effects have to be permanent. When you make a brain cell, it has to stop being able to do anything else. So a permanent tag has to go on there. And I'll show you later on what makes that permanent. So there are 30 million points along the DNA where tags can be added by writers, read by readers, and removed by erasers. The tails are a string of chemicals. And there are locations in the tail noted by K, that's lysine. Oops, sorry. And that's where these tags can be attached. You can attach 2 tags or 1 tag. And these tags have different locations. And at these different locations, they have different effects. So at one place, you could have 1 or 2 tags. One of them creates an activity. It creates the ability to produce a protein. And if there are 2 tags, that enhances the protein producing activity. Here's one that silences a particular protein creating capability. Here's one that represses a polycomb protein creating capability, and so on. This is called methylation. This is called methylation. Whenever you add a tag, you're methylating the tag. And if you remove a tag, that's called demethylation. Each histone tail has its own unique structure. So there's the 4 types of histones, and they each have a different kind of tail. And each tail has different locations where it can be methylated. And some of them have tags called acetylation tags or phosphorylation tags, and so on. So there are different tag sites and different tag types. Before epigenetics was understood, back before 1972, variations in siblings that could not be explained, and actually variations in a creature itself, they couldn't be explained. They were called intangible variation. An example of this is a caterpillar and a butterfly. They have exactly the same DNA. They're completely different. This is an example of episodic epigenetics. And scientists didn't have any way of explaining what triggered this change or how the change worked. So they just put the label on it. Oh, it's an intangible variation. Here's an example. The gender of a crocodile, something I never knew, is determined by the temperature at which the eggs develop. Well, that's an environmental trigger, that the external environment of the egg affects how the egg develops. So somehow the environment is triggering the epigenetics of the egg to change its direction of development to become either male or female. The DNA of a queen bee is the same DNA as that of a worker bee, but she lives 20 times longer. She's much larger. The difference is she has a diet of royal jelly. So here's another example of an environmental impact outside the egg that causes the egg to develop an entirely different way. And this is true of plants. When does a plant come up out of the ground? How is the sex of an amphibian created? The caste and social insects, the coat color of a mouse. We now recognize that all of these intangible variations are epigenetic phenomena. So it's the epigenomic mechanism that triggers the expression of one or more specific genes during blastocyst development, during puberty, during egg fertilization, during pregnancy. How was this discovered? There's a laboratory mouse called an agouti mouse. The agouti mouse was bred for the study of a particular gene called the agouti gene. That's why they called the mouse the agouti mouse. In addition to making the rodents different from other mice, it made them bigger. It made them very hungry, ravenous. It made them yellow, and it also made them prone to cancer and diabetes. Now, why did they breed these mice to be different from normal mice? So they could study that gene, and so they could study why that gene made them prone to cancer and diabetes. So if you want to find a cure for diabetes, you get yourself agouti mice, and you try to cure their diabetes and try to figure out what's causing it. Well, one of the things that happened in the research of these agouti mice, and this happened accidentally, a cage full of agouti mice was fed incorrectly. And because they were fed incorrectly, when they had their pups, a bunch of them were smaller and looked normal. They were half the size. They were a different color. And nobody could figure out why, but they figured it must have something to do with the difference in the way they were being fed. And that led to the research that led to epigenetics. On the smaller mouse, it turns out, there's a methyl group, a tag made of hydrogen and oxygen. That's what a methyl is. And it becomes affixed to a tail on a histone in the agouti mouse's gene that shuts it down. And when it shuts it down, it inhibits the gene from activating the DNA that causes the mouse to not gain weight. So having the tag inhibits the gene, the mouse doesn't become fat and diabetic. So that's what methylation is. Methylation affects the agouti gene in the mouse. It affects the color, the obesity, and its vulnerability to other diseases. So methylating it gives it the yellow, the fatness, the disease. Demethylation cures it. There's another kind of tag called an acetylation tag. Acetylation tag affects how tightly the DNA is coiled. Now remember, RNA has to come in and read a string of DNA that's between two histones. Well, if the histones are right next to each other, the RNA can't even get in there. So acetylation tags close that gap and prevent the RNA from coming in and reading the gene, so no gene expression. And the acetylation tags can actually make it tighter or make it looser. So it can shut it off, or it can just make it lower, so you can produce less testosterone or no testosterone. Or you can loosen it and make it generate more testosterone. So here's an actual microphotograph of very tightly wound DNA, right? This is called a silent chromosome. It's condensed, it's closed. All of these genes are turned off. But here's the same microphotograph of an active piece of chromatin. It's decondensed, it's open, the gap can be read by RNA, so this little gene in here can be read and produce a protein. So this is turned on. What this is a picture of two genes on the X chromosomes of a female. A female has two X chromosomes. A male has an X and a Y. Because a female has two X chromosomes, one of them has to be turned off in every cell. And this is how it's turned off. And one of them is canceled, and that opens up one of the X chromosomes, but the other is closed off. Genetic control via epigenetics is a normal mechanism. It starts and stops developmental process. It differentiates 200 cell types for 70 trillion cells in your body, and enables responding to the environment faster than evolution allows. That set of tags is called the epigenome. So what impacts the epigenome? Well, one thing is where the cell is in the organism, because when a cell changes, it produces chemicals that are actually shared across the substrate with other cells that are nearby. So if a cell is between a bunch of brain cells, it's going to be influenced by the genetics of all the other cells to become a brain cell as well. Where the organism is in its developmental stage is going to impact what genes are being expressed and which are not. And environmental factors are going to come in from outside and impact the epigenome. Some of these things from outside are food, vitamins, caustic substances, and stress, because stress produces hormones, and the hormones impact your epigenetics. In the 1980s, scientists studied the birth and death records of an area in 19th century Sweden called Norrbotten, and people that lived during the times of want in Norrbotten lived on average 6 years longer than those that lived in times of plenty, but they were overweight primarily. They were typically susceptible to metabolic disease like type 2 diabetes and obesity, and they were more prone to having mental health issues. And amazingly, their children were the same, and their grandchildren were the same. That meant that epigenetic difference was being passed on even after food was plentiful. Women who were pregnant during a 1944 famine in the Netherlands, known as the Dutch hunger winter, had children and grandchildren who were unusually small or prone to diabetes and obesity, an epigenetic change. In 1998, Quebec suffered a major ice storm. Power was lost for months at a time. The children of pregnant mothers from this time were followed for several years, and the children developed more autism, more metabolic diseases, and more autoimmune diseases. So that was an epigenetic change that was passed on. Nutrition can impact your epigenome. A female agouti mouse fed a diet rich in methyl groups has pups that are not typical agouti mice. They're small, they're brown, and they don't have obesity, and they're not prone to being diabetic. And their offspring are normal mice as well. When cells divide, the epigenetic markers are copied by readers and writers. So a liver cell divides, readers read the tags in the mother liver cell, and then as it divides, writers have to replicate the tags on both sets. So both of the new cells are liver cells because they have exactly the same tags. But when zygotes create a gamete, right, when a sperm meets an egg and it turns into the start of a new being, these markers may or may not be cleared back to their base state. So when zygotes create a gamete, there has to be some mechanism that clears some, but not all, of the tags. And what we find is that that is actually what goes on. Methylation starts out high at the beginning, But after fertilization, methylation goes down and the tags, for the most part, start to get removed. So the epigenetic tags from the father's sperm are more aggressively scrubbed than those from the egg. And there are also some imprinted genes where only one copy is ever read. So those are protected from scrubbing. So some methylation tags do get passed on. Also, the egg contains tagged strands of DNA already. So they're not changed by conception at all. Researchers gave pregnant mice chow containing only half the calories they needed during the last week of gestation, a time when the epigenetic patterns in a male embryo's primordial sperm are erased, then reset. And this treatment resulted in offspring and grandchildren that were underweight and prone to diabetes. So being deprived of calories had an impact. So if you're pregnant, depriving yourself of calories is a way of hurting your offspring and your grandchildren as well. And here's a real life example. There's a woman, Sonia. She lived in Kiev, and she was pregnant with a girl back in 1915. The girl, fetus, her name was Dinah, included all the eggs she could ever carry. When a fetus is formed, it's a female fetus, she's formed including all the eggs she's ever going to carry. And Dinah and her eggs were affected epigenetically by Sonia's environment during the development of Dinah. So whatever Sonia did she smoke? Did she get enough folic acid when she was pregnant? Was her diet good? Was she subjected to a lot of stress? Whatever that did, also, whatever she did affected Dinah's development. Then later, Dinah becomes pregnant with a girl in 1948 from one of those eggs that was formed in 1915 and was also impacted by her mother's behavior. The egg at that time was 33 years old. And that egg and fetus were further affected by Dinah's environment. How was her diet? Did she smoke? Did she get enough folic acid when she was pregnant? Was she subjected to stress? If Dinah made good choices about diet, vitamins, drugs, alcohol, tobacco, stress, and so on, this might change tags that were created in the eggs that Sonia made that might be in the fetus that Dinah is carrying. Then that baby was born in 1949. Her egg age at the time she was born was 34, and her epigenome at the time of birth now includes the effects of her grandmother's and her mother's environment and lifestyle choices. When the cells divide, they create a gamete. These markers may or may not be cleared back to their normal state, and if they don't, they become an inherited epigenetic trait. The addition of environmental toxins to the diet of an agouti mouse can cause the epigenetic change to disappear. That's called demethylation. So feeding alcohol to a pregnant agouti mouse will cause it to have a different ratio of methylated and demethylated pups. Feeding a fruit fly, a male fruit fly, dietary sugar before it impregnates the female will cause obesity in the offspring. This is also true of mice and very likely humans. So a male parent eating a lot of sugar can result in obesity in the children. Stably heritable phenotype resulting from changes in a chromosome without alterations in the DNA sequence, that's what epigenetics is. A cytosine tag turns the gene on and off. That's methylation. Acetylation tightens up or loosens the space between the nucleosomes. There's another piece, miRNA, that's messenger RNA inhibitor, can actually occupy the space between the histones and block the RNA from being transcribed into a protein. In addition to these mechanisms, there are more than 80 plus chemical modifications that can occur on a histone tail. So this code that programs when a gene is turned on, when a gene is turned off, how many genes are turned on, how much they create in terms of proteins, is very complex. And whoever figures out this code, and I suspect it will be artificial intelligence that breaks it, but whoever comes up with this is bound to win a Nobel Prize, because if you understand how epigenetics, the code that programs your DNA to behave in a particular way, will then be able to program DNA to behave in any way they want. Identical twins have identical genes. They're clones. They should then have similar expressions, and they do. And identical twins also have similar body language, and they do. And they should have similar abilities, and they do. They're clones. But one might get cancer and the other might not. One might have schizophrenia, and the other will only have a 50% chance of getting schizophrenia. In addition to epigenomic changes, identical twins may accumulate somatic DNA mutations as they age that make their DNA not completely identical. There are many diseases that are impacted or caused by epigenetic dysregulation. So in a study, genetic material was taken from 40 pairs of identical twins and it was examined. And when it's examined, what they do is they compare two strands chemically, and if they're identical, then it shows up as yellow. And if they're not identical, it shows up that they're different. And what they saw was that when the identical twins were 6 years old, they were nearly identical in terms of gene expression. In other words, their epigenetics were pretty much identical. But when they were 66 years old, 60 years later, it was very different in how they were expressed. As identical twins age, their epigenomes change based on lifestyle and exposure. So, diet, exercise, exposure to chemicals, smoking, what kind of medication they take, stress, all can lead to the suppression or hyperexpression of proteins. An example of this is shown when monkeys are raised by a nurse. So monkeys from the same, what do you call?
Offspring?
Right?
Offspring or something like that?
Well, a collection of pups, right? So monkeys have a set of babies, and half of them are taken away and raised by a nurse. So they're bottle fed,
that's the word,
as opposed to being nursed and stroked by their mother. And the ones that are bottle fed by a nurse are more aggressive, they're more stressed, and they have a higher propensity to become alcoholics when fed alcohol. And when their epigenome is mapped, showing methylation and demethylation, they are entirely different than the ones raised by their mothers. Example of this is, here's a mother rat licking its offspring. When the mother rat licks its offspring, the brain of the pup produces serotonin, and the serotonin blocks a glucocorticoid receptor, and blocking that receptor makes an epigenetic change that makes the pup less stressed and more able to handle stress for the rest of its life. If the rat is not licked, the serotonin is not produced, and the rat can't deal with stress as well for the rest of its life. It also changes their sexual behavior. So lack of a motherly attention in an infant's life is going to have an impact on that child for the rest of its life. if it changes the methylation or demethylation of certain tags in the epigenome. Affection creates serotonin. The serotonin has a specific epigenetic effect on a tag that later impacts the pups as adults. Aristotle famously said, "Give me a child until he is 7, and I will show you the man." Because as a child, you can give the child love and affection, or you can ignore the child or abuse the child. You can give it nourishing food, or you can feed it junk food. You can give it a safe environment and make it feel secure, or you can make his life a nightmare. Or you can give him a clean environment or expose him to secondhand smoke. So what you do as a parent is going to not only affect your child, but maybe your grandchildren as well. Children who are abused and neglected tend to be alcoholics, addicts, more suicidal, and have mental illnesses and be socially maladjusted. So epigenetics is impacted by how much you're touched as an infant, how much you're encouraged as an infant, how many hours of sunlight your mother was exposed to, how much nourishment was available, how clean the water was, how clean the air was, how often you were frightened as a child, how badly you were frightened, and for how long you were frightened, and how much exercise you got, and what kind of exercise you got. So there's a very famous question. Are you the way you are because of nature or because of nurture? What are the effects of being born impoverished on weight gain, on emotional quotient, on intelligence quotient, on your ability to have relationships, on your ability to be a good parent? It all depends in large part on your epigenome, what switches are thrown, how the dials are set from the time you're born, and how those dials and switches are reset as you age. So is it nature? Yeah. If it's your DNA that did it, it's nature. Is it nurture? If it's your epigenome that's doing it, it's nurture. So unlike mutations in DNA, epigenetic mistakes are reversible. And that means that we could produce drugs that could reverse bad methylation or bad demethylation in your epigenetics. Now, there are diseases that are caused by gene mutations. They're called monogenic diseases, and they could potentially be cured by using technologies like CRISPR to actively edit a particular gene to either not produce or to produce a particular protein and cure that disease. And there's actually been work done there where those cures have been created. Most disease mutations, though, occur outside of genes, which means they're epigenetic in nature, and they're also polygenic. When a gene produces a protein, that protein could trigger epigenetic changes, tags, on other genes in the DNA. And that can result in diseases like diabetes, schizophrenia, multiple sclerosis, and cancer. Now, rats have been made to become cocaine addicts. I mean, the things we do to rats and mice in laboratories is terrible. But if you make a rat a cocaine addict, it will self-administer cocaine until it dies. So here's a picture of a rat. It's got a probe in its brain that feeds it cocaine as long as it steps on that lever. So the rat figures out if it steps on the lever, it gets a dose of cocaine, and it steps on that lever until it passes out. And when it wakes up, it steps on the lever again, and it keeps doing this until it dies. But increasing methylation increases the desire for the drug, and decreasing methylation cures the rat of its addiction after a single treatment. So even drug addiction can be cured by modifying the effects of your epigenome. Now, not all diseases are curable or caused by epigenetics, but those that are may be curable through epigenetic reprogramming. In 2009, research was done that found that some children with autism had extra DNA referees. I'll tell you what those are in a minute. And turning off a gene needed to respond to oxytocin. And that's a hormone important in social interaction. So the study was small, but it suggests that turning off that one gene could cause the social problems people with autism had. Until recently, scientists thought genetic mutations played a bigger role in cancer than epigenetic changes. But now we know epigenetics plays either an equal or a bigger role in cancer than genetic mutations. There are tumor suppressor genes that protect cells from becoming cancerous. The epigenetic tags can turn these genes off, stop the suppression, and cause you to have cancer. Drugs can target these genes to remove, or these tags rather, to remove them, allowing the tumor suppressor genes to perform their function. The first epigenetic cure was in 2006. It was a lung cancer cure. It was a pill. It was taken orally, and you can see in the pictures that in 8 weeks of treatment, the tumor went from the one on the left, which was pretty massive and solid, to one that was smaller and emptier. And this was pretty much a cure of a particular kind of lung cancer in only 8 weeks using epigenetic manipulation. The primary principle behind this therapy is targeting epigenetic changes with drugs that reverse damage and return the expression of DNA back to its precancerous condition. And these drugs are being developed. They've been approved by the U.S. Food and Drug Administration to treat ER-positive metastatic breast cancer, lymphoma, myeloma, precursor to leukemia, and other diseases. And they're constantly working on cures for cancer, rheumatoid arthritis, type 2 diabetes, addiction, asthma, lupus, obesity, Alzheimer's, Parkinson's, autism, schizophrenia, bipolar disorder, and Huntington's disease. Now, normally, as you go through life, you remember the epigenetic tags change. And if we look at those tags, we can determine by looking at those tags how old you are within 2 years, plus or minus. So if we look at your tags and they look like this, we can say you're between 58 and 62 years old, and it's very accurate. And what we've learned from this epigenetic clock is that children age epigenetically much faster than adults. And this makes sense because children's epigenomes have to be modified very rapidly during their development stage. Their brains are changing, their ears are changing, their eyes are changing, their bones are changing, everything is changing. And it happens very rapidly in the first 2 years, and then it slows down. Men age faster than women. Aging signs are impacted by life exposure and lifestyle. Chronic stress accelerates aging, but strangely, acute stress that's short-term does not. And cancers age faster than normal tissue. 66% of all deaths today are attributed to chronic diseases, which are epigenetic. 75% of healthcare cost is attributed to chronic disease, epigenetic. Disease often involves the dysregulation of normal gene expression. Cell types can forget what they are due to epigenetic changes or mutations to DNA. Broad opening of chromatin due to a loss of histones. Or remember those loops? If a loop detaches from the wall that it was normally attached to, it's no longer the two loops. When it became one loop, those two loops are no longer separated, and changing the epigenetics on one side of the loop can now affect epigenetics on the other side of the loop. A mutation among the readers, writers, and erasers that manipulate the tags, that can cause problems. So we can create drugs, but remember, it's really complicated. The drugs have to be the right drug, and it's got to find the right tag to hit the right location on the right histone tail of a particular gene. So it's not easy stuff to do. The efficacy of the mitigation of aging-related issues can be tested, though, using the epigenetic clock. So if we look again at the epigenetic clock, the blue line is a normal person. The red line is what we see in the epigenome of HIV-positive people or people with Down syndrome. And you notice their epigenetic age is higher. The green line is the offspring of people that live to be over 100. And you see their epigenetic age is lower. So that's why they can live longer. So we can create a drug and see where do you lie on this line, which line do you lie on, and we can see if those drugs are good or bad, and are they effective. Now you can affect them yourself. And what can you do? Well, remember I mentioned referees? Referees are the regulatory network. It's a feedback loop. So a stimulus comes in, a particular gene is triggered. The gene produces a protein. The protein triggers another gene. That protein, that gene rather, creates another protein. And that protein triggers the stimulus protein that started this loop. So it goes round and around, and it also continues to do whatever function was being performed by this gene in the first place. So the gene actually had two functions. One was to turn your eyes blue, and the other one was to come back and say, and it's always going to be blue, and it keeps rotating through. There are more complex situations where you have more than one gene that's involved in the effect you're going for. So we want to turn one gene on maybe and one gene off. So if you turn this gene on, it goes on to perform its function, but it also feeds back to stop the other gene from ever getting turned on. And that creates a permanent situation there. Now, because this is a constantly ongoing loop, that means the methylation and demethylation doesn't stop. It's an active process. So you have to have the ingredients that you need to do methylation. You got to have betaine, choline, methionine, homocysteine, methyl, and so on. So there are things you can do that accelerate epigenetic aging. That's a bad thing, right? So getting your vitamin E from soybean and corn oil, bad, accelerates epigenetic aging. Having high sugar levels in your blood accelerates epigenetic aging. High triglycerides is bad. So that's animal fats, right? High insulin from being a type 2 diabetic accelerates epigenetic aging. Having high blood pressure, having a high body mass index is bad. So don't be fat, be thin. Processed foods, food coloring, pesticides, air pollution accelerates epigenetic aging. Mothers smoking during pregnancy affects their children's epigenomes and tends to increase the probability of cancer and the development of cancer. There are 50,000 genetic variations that are possible results of smoke and secondhand smoke. In a 2009 paper, 12 people who were abused as children and later committed suicide have different DNA referees on a gene needed to cope with stress. So they couldn't cope with stress. That was an epigenetic change. Endocrine disrupting chemicals called obesogens tend to make you very, very fat. And they're found in anti-fouling paint, but that's only if you're, a sailor and you come in contact with that. On another thing called dibutylin, which comes from hard plastics. And it's also a chemical used to deworm poultry. So depending on how the chickens you eat were raised, you might be exposed to this. Bisphenol A is a very common plastic that is found in water, in microplastics in water. It comes from hard plastics that get dissolved in the ocean, so it's pretty much everywhere. But eating folic acid counteracts the negative effects of bisphenol A. So taking folic acid, can help protect you from epigenetic factors. So get that in your diet. Other things you can do with your diet to slow epigenetic aging. Eat poultry, fish, fruits, and vegetables, not red meat. And fish has the greatest impact. So if you can become a pescatarian and live with it, that's a good thing to do. Eat carrots, sweet potatoes, plants with green leaves. Coffee and chocolate are pretty good for you in terms of epigenetics. Vitamin E from olive oil is good. A moderate amount of alcohol, and especially wine, is good for you. Getting folic acid from broccoli and beans is good for you. Eating broccoli and garlic is good for you in terms of epigenetics. Brussels sprouts, kale, and B vitamins, very important. Now, if you're going to be a pescatarian or a vegetarian, you're going to lack vitamin B12. So taking a vitamin B12 supplement is very important. So that's it. That's epigenetics, what it is, how it works, and what you can do about it, and what can happen to you if you do nothing about it. So thank you for listening, and that's the end of the talk. And if you have any questions, now's the time. So I'm going to stop sharing now.
Mm-hmm. You can keep the sharing as well, because nobody has videos, and it's just nice to have your, this Vitruvian man as a signal.
Well, okay. Anybody have any questions or comments or anything?
Yes, I have a question.
Yes?
About the folic acid. Does it concern only on pregnant women or everyone?
Well, it's everyone. I take folate every day because if you're exposed to something that messes with your epigenome, folic acid will actually correct it.
Yes, but a few days ago, I listened to a lecture of a Russian famous doctor. She said that folic acid can cause cancer.
I didn't hear what you said.
Cause cancer.
The doctor said what?
Sorry. Let me just handle it.
1-2-1-2 cancer. She says cancer.
Oh, really?
Yeah, something like that. What was the name of the doctor, by the way, Milan?
Well, there's a link. There's a link on the slide that will take you to where I read that. So you can look it up and see if you agree or not.
Okay, thank you.
Yeah. I love Ron for how thorough his lectures are, and if you pay attention, like sometimes when I see your lectures, I don't notice, but there are like whenever I just notice that at the bottom of every list in this big slideshow, you always show the original paper where you reference or something you have to go and check for yourself. So this is really an academic approach that I appreciate, because this one lecture that is 2 hours or like 1 hour and a half mostly, it's just precious, because you can always go back to it and listen again and go read those books that you mentioned, or maybe some articles that you mentioned too. This is really this is how we educate, I think, in this new era. My beautiful background. So yeah, I think this is how we educate and share information, how we use our critical thinking to actually, help yourself decide what's going to be the best way to live your life, and to, it's back. Okay, so it's funny because I have this picture, I copied it and I have it in my house. So some people think it's my real background. So yeah, I think this is so amazing that you are a professor, you're much older, as I respect you just as an older, a person who has more experience in gathering information, training, critical thinking, having life experiences, families, and many decisions that we can analyze. Because when people ask me like, what do you think about this thing or maybe that thing in your life? I say, I cannot judge. It's really hard for me because as in science, they say we stand on the shoulders of giants, right? So this is really cool that when you take a moment to appreciate that, basis of information that you have today and to be able to add to that. So I think we have to do this as, citizens of the earth, that we have to educate ourselves, share knowledge, support each other, help each other, and become the new population of the emerging planet as a collective, hive mind species.
Yeah, I think university type lectures should be done this way. I mean, universities could be almost free if this is the way we did it. Right?
Look at Chomsky. When I really appreciated a couple of his lectures in Arizona University, I heard the audible, I think, but I think there's also a video of him being rather old, and he shares his opinion about education. So that's just a Googleable, YouTube video about education from Noam Chomsky. It's basically a lecture, and he says so many things that I think, wow, it's so difficult to notice the thing that is too close to your face. And when you get a little bit far away from that, that's the only focal point where you can actually figure out what's going on. And that's why I love, theory of everything, because I live on a different, part of the planet, on a different part of the spectrum. And that sometimes allows me to highlight some of the differences and pinpoint those places where people collectively make mistakes and do not notice, they do not use their critical thinking to change the world to the better. So there are many questions like, do they not see it? Do they see it but ignore it? Or maybe do they, are they comfortable with that? So yeah, many questions.
Well, they're too busy following their favorite soccer team.
I guess so, or using Instagram.
Yeah.
That's the saddest part of, well, at the same time, I think we live in a very exciting moment in history where education is being reborn, I would say. I really want to try in my, language practice to have these Schola formats. I am really interested in learning more about ancient worlds. Like, I was so fascinated by the HBO series. Like, there are 2 seasons. Rome, it's called Rome, so it's really very explicit. It starts with a war by Gaius Julius Caesar, and like how this whole story is going. But that's not the point. The point is that you see human society at its best, and what things were wrong in that civilization and what things were good in those civilizations. We have to really go back and check out our chests with all those ancient manuscripts and check out our revisit our history, because we have to really slow down in our progress and work on our social and survival strategy. Basically, it's straightforward survival, because either AI, sentient aliens of any kind, positive, negative, influence, maybe some of them are gonna be really aggressive. Are we ready for that? Maybe some of them are gonna be so advanced that they could just be a danger or a challenge to humanity. Are we ready for that? I don't think so.
Well,
yeah.
I think the aliens we have to worry about most, though, are artificial intelligence.
It's closer. Yeah, I think artificial intelligence.
Yeah,
and design.
Very close.
Yeah, it is here. I'll tell you that. In 2020, when I went to study at the university, well, I was surprised actually. It's an official certified university on a territory of Russian Federation, which was called University of Artificial Intelligence. I'm like, wow, this is so cool that they took this name, which is bright and flashy. And every time people ask me, what are you doing on your COVID, isolation, I would say I'm studying artificial intelligence, and I learned coding and all that. That was so hard, but I didn't manage that. But it's been 3 years ago, and we we saw in details how artificial intelligence works. I actually have a lecture that I used for speaking a competition that they had inside university because they were looking for new lecturers, and narration something, and I was explaining how neural networks, computer neural networks work. I explained how the brain is functioning and how we build mathematical, computer structures that were based on our brain, like simple neural networks, and how you play around with them. So since then, in 3 years, the progress was so swift that I realized that the faster than I graduate and acquire this profession for what I want, because my motivation was to have fun and learn to code, to create my own assistant, for example, right? Something that is going to help me. For example, I'm not very good at singing, I'm not very good at dancing, but I can speak and maybe come up with stories or combine somebody else's stories. How do I acquire a helper who's going to illustrate stories? You could take legends, you could take, any text, which is going to be like 5 pages to something. Welcome to ChatGPT. And like you see, all these instruments are, the fact was that AI is developing faster than a human brain is, able to acquire this instrument and harness it. Even those groups of specialists who are present in the world at the moment, 3 years, I see the difference in what a leap artificial intelligence has done. We have no regulation for that. We only started, like, the Senate had a meeting with IBM specialist, OpenAI director, what was his name? Sam something, and another person. Like, these were big companies in tech, and they were discussing artificial intelligence. And some of the Senates, they were asking questions and discussing, so they eventually decided to create a body, like an institution that is going to control that and oversee what's going on in artificial intelligence. There are like 4 models like ChatGPT. They're very quick and they work, they show their effectiveness. These are not experiments anymore. These are, persistent birth of a new synthetic sentient being. We have to be ready for that. This is a mirror, as I say, this is a mirror. What we create is a mirror of us. If we are vicious and we see the world as a dark place where we have to be those traitors who, can stab somebody in the back just to survive, right? What was the abbreviation? VOCAFC something?
Viability of current and future generations.
Yes. Yeah, egoistic, selfish gene, as was it Dawkins who wrote this book? Like, the selfish gene, it's ego, like, maybe you can call it, or maybe the dark part of us, or maybe a shadow personality, if you ask Jung, for example. This is something that protects us. These are our DNA, basically, that once is a machinery to protect itself and calculates what's going to be the price, right, or the worthiness of any act. Like, having brain is very expensive acquisition. It weighs just 2% of our overall weight, but it consumes like 20% of our energy overall. It's a very expensive, powerful machine, and I'm not sure that we are using it in the right way. I think we're using some, a laser gun to nail something into the wall, right? So nail a picture into the wall. So because we don't know how it works, the laser beam, like maybe swords from, Star Wars, something like that. We don't know how to use that. We've seen only axes and hammers, so we use it as a hammer, right?
Well, we have a lot of evolutionary baggage. Mm-hmm.
Let's talk
your
about
brain.
that.
Your brain evolved in a world that no longer exists. Mm-hmm. So a lot of the things it does are there to protect us from saber-toothed tigers and distract us from what we're doing when a shiny object moves in the bush, and so on. And we're easily manipulated. We're easily manipulated by people who study how to manipulate us. And all it takes is, if you were a very, very rich person and you could hire psychologists
to manipulate,
who know how to, who know how to manipulate people,
public opinion,
then
like 76%
you can manipulate opinion, you can manipulate behavior, and we are subject to that. And that's what social media is doing to us now, because that's all designed to be addictive, for example.
Yeah, I'm guilty. I am guilty. It changes. Another aspect that I don't hear about, I have no research on that. How does this new digital synthetic, part of our civilization, it's really important to pay attention to that. But we do not pay enough attention to what's going on between these, synthetic life and organic life. I think we remember about how the tendency with psychological diseases is changing. In the past, people may be underdiagnosed, but we see that even in our newest history. Like, I watched the lectures by Robert Sapolsky from 2010. It's been on YouTube since 2010. It's been 13 years. And he says, in the future, by year 2020, he says like that the percentage of people who are going to be who have psychological problems, they're going to double or something like that, become a fourth most crippling, disabling disease in the USA, something like that. The numbers were crazy. Like, third number was diabetes. And then the rest is going to be something. Who is Robert? Robert? Who Robert? Let me check the message.
His first name.
I have one of his lectures in one of my presentations.
Yeah.
Yeah.
Sapolsky. He has obviously he has Polish background, but he lives in America. He's a scientist. He's like a very interesting bearded guy. Like, he worked with a lot of primates and apes, and that's why he has very famous pictures when he is wearing some kind of safari outfit with baboons and marmoset monkeys, and he tells very interesting lectures, and he just has very interesting personality as a teacher. I really enjoy his lectures as well. The way he interacts with young people is just fascinating for me as a teacher.
Yeah, very good. He's an evolutionary evolutionary psychologist, basically.
Anthropologist, neurologist, even. I was surprised. I'm like, is he operating or anything?
But I mean, the main thrust of it is the reason you behave the way you behave is because of how you evolved.
Yeah, human biology of human behavior. Biology. Yeah. That was the name of 10 or 25 lectures from Stanford University. Biology of human behavior. Well, actually, if you Google his name, if you use his name on YouTube, you'll find it very easily. Chernigovskaya, I have a lot of questions about Chernigovskaya, I'm gonna tell you. It's like a famous woman, like, it's gonna be fun if you watch this video. It's like, I really wonder what other people from other cultures who have no exposure to Russia whatsoever, how do they feel watching something for the first time in Russian with subtitles, like just to get a ticket into this world where it's isolated totally because of the language. Russian language, languages form your consciousness, like they shape who you are, because different languages have very different yeah, it's like they sound very differently. There are simple examples, like we say in Russian, we say v internet, in internet, not on internet. So we see information as a cave where it's cold and scary, and you get into it and you it's like you, you're crawling around a cave or something like that. So, but in English, I explain to Russian speakers the difference in thinking. I say, well, in America, they say on the internet because you surf on the internet. So for you, it's a sea of information, and you surf on the surface, which is cool, right? Everybody likes surfing. And, being in a cave is a totally different, thinking and totally different feeling. Because, for example, there are a lot of interesting cultural differences. in different languages, because I am a native, I'm native in 2 languages, so I'm bilingual by birth, and then I started learning English when I was like 5 something, and I've been studying it, and then when I was 20, I had like enough experience to, and a lot of exposure to native speakers, so when I went to America, I felt very natural, that like, as if it's my 3rd personality, basically. So it's really...
She's not just a lexicologist, she says she's a neurologist.
Neurology. Yeah. Chernigovskaya is definitely curious, but I have to share my opinion with you, because after I watched more videos with her and I started, pulling that thread out, I realized that she's not a very strong academic. She speaks about linguistics and English. I have never heard her speak in English, ever. I took her lectures, like a course of lectures on Coursera, where the whole lecture was like maybe 10 or 15 lectures, like serious lectures. You had to pass the tests and all of that. A couple of my colleagues, they also passed this neurolinguistics course from St. Petersburg University. She's an administrative person. That's definitely administrative. So she is at that institute. She's somewhere.
She has strong ties, yeah.
Yeah, but when I went to Google, that was I didn't finish the story, that the text of her lecture was in English. The slideshow she's using to explain stuff is in English, and she's using a Russian language where it's really confusing, because she tells you in Russian language some part of the brain, for example, and you see the signatures and the legends in English, and you're like, this, yeah. And
it's okay for Russian academical lecturers and teachers. It's okay for us.
I don't see that as professionals. Yes.
What other people think, and especially from the US, what they think who she is, because there are some people who think that she's even a psycho, she's a schizophrenic person.
I think that a lot of people respect her because she looks like a grandma who's always right. She has like this snobbish, character, which is, I think, very likable for many Russians. They see her as like a grandma who's going to tell you whatever. Okay, so it's just interesting. If you ever have time, some free time to waste, YouTube something about Chernigovskaya. That's her name. This chat is gonna stay with you guys. So Chernigovskaya, she's a neurolinguist in Russia, and she talks a lot about brain, and that she's a little bit too conservative. So I don't know how she combines these two. So she's like a public, science speaker. So she talks a lot about that. So, mixed feelings about her anyway. So coming back to epigenetics, do you have any other questions, Mylin? Do you have any questions?
Yes, I do.
Yes.
Ron, I have a question about detecting this situation, and I see this kind of issue with my parents as well, and it's mostly dietary, and now I'm seeing it's epigenetics as well. And regarding the diet you mentioned, where how these issues could be detected first, and second, for the diet, I've seen one specific statement of the diet that plants all the time has a defending chemicals all the time to survive, and it's evolutionary, so all the time fruits can be properly eaten, but the plant or the main structure will be all the time defended by their chemicals to be avoided to be eaten by animals or any environmental.
Right. But don't forget, we've been cultivating plants for more than 11,000 years, and they are very different now than they evolved to be. So, those that we eat for the most part have the bad effects bred out of them. A banana today is not a banana from 10,000 years ago. They look entirely different. Eggplant is
they used to be green and like not, so sweet. I think they just selected them.
They were very small and full of seeds, and they're not like that anymore.
In my view, banana is still helpful because it's fruit. Right now, what I'm saying is that fruits all the time better
right.
to eat
They're
for
always
a long
friendly
time.
because they propagate
Yeah.
by being eaten. But
Yeah.
but like I say, we've been cultivating green plants and corn, and it's all very different from what it used to be.
But where's the seeds
But yeah, there
and
are
seed
foods you
oil?
shouldn't eat. There are foods that are very good for you. Don't forget, if you were here for my microbiome talk, a lot of beans, for example, have fiber, and fiber is necessary, and
But
you
long term,
have to eat
what
it.
do you think about the eating every day? Like it has a little bit of high damage. Like,
Well, the best things to eat are what's known as the Mediterranean diet. Even if you look at what I said you should eat in this talk, it's all basically the Mediterranean diet. And if you look at the longevity of people that live on the Mediterranean diet, they live longer than people that do not. So clearly, it's doing the right things.
Can you see my demonstration? I when you spoke about the like domestication of different species and plants included, like selection has changed wheat and rice so much. Like wild wheat, it looks like this, like on the left, and red wheat that we used later, domesticated variety. It's like we selected them for being much more dense and larger and certain properties.
And now they're being genetically modified. So if there are any chemicals in there that are undesirable, they can actually change the genetics of the plant and get rid of those chemicals.
Oh, Milana was asking something, and I see her questions in chat. Do ever only singular, not systematic moments of stress affect on the epigenome? And does the stressful year for us have a chance to affect on our future, children's eggs?
Yeah, the longer you're under stress, the greater an effect it has. Short-term high stress does not affect you as much as long periods of stress. So if you grow up in a war zone and the war goes on for 20 years, like in Afghanistan, well, they've been at war for 500 years, right? The people there epigenetically must be a mess, because I don't know how they could possibly deal with that. Or if you grow up in an area that's constantly having droughts and famines. In fact, there's a place in Africa where they have a famine every year based on the seasons. And the epigenetics of the people that are born during the summer are totally different than the people that are born during the winter.
Oh, my favorite, set of research papers. I was trying to find the lecture that I had. I had a piece of the magazine that talked about the research by Dr. Kyriazis. I don't remember from which university, but he researched like different sorts of stress. And for example, if you're nervous in the moment, for example, because you're late for an airplane, and having to take care of your dying mother who's going to be, experience her Alzheimer or dementia, these are two different types of stress. The first one, even if somebody dies, but it's like not dies, but it's like something like a car is going to hit you and you evade, the immediate death, but basically like in half a second or something like that. That's also a very strong stress. Like there's fear, there's a lot of
serotonin.
Like a lot of different emotions. But chronic stress, which is I hate my job. I'm not happy with my family, or maybe I have to take care of somebody who is, it's not going to disappear in years, like 10 years. That's a whole different kind of stress. And being around a person that you don't like entirely for a long time, that's a chronic stress. So these are, short term is good.
There are some researches about, curse words. Which mothers or pregnant women say, and they affect on their future children's psychology. Does it have some ties with reality?
Linguistic curse? Linguistic abuse?
Well, как это чувство, the emotions you hide in them in the curse words, they really affect on your future children.
That's a good question. What if you feel that stress about something and you are not allowed to scream? When you have to suppress your emotions, but you're not gonna hide it from another little body that is very sensitive and is bound into your system. Yeah, maybe linguistic imprint takes place, but I think there are many other factors. It's more about what you feel. Cortisone and like all those mixture of different hormones that you experience, they're gonna be in the bloodstream, and children are going to feel that. I really wanted to also mention some of the branches of interesting stuff that we could explore. I really enjoy Stanislav Grof, because I'm a linguist and I enjoy how he speaks, because he's from Czechoslovakia and he's like over 80, 90 years old. I hope he's still alive. I don't really remember. Stanislav Grof, yes. Stan Grof, he has a very rich, interesting history of medical practice. He was a medical student and started working where he got into a group of people who were going to test some new substance that was invented in Sandoz company in Switzerland, which was called LSD. So he was one of those people who experienced that, and then at early age, and he had like a whole long line of different research papers and academic work, and eventually he got into like a whole new area where he had some trainings for people to have. He invented a breathing technique which is called holotropic breath work that imitates the effect of LSD that he has been researching for a long time. And they found he was one of the participants and founders of transpersonal psychology, which is the main. They believe in previous lives, first of all, because there are a lot of interesting, witnesses from Buddhism, for example, like how they choose the next Buddha, or like Dalai Lama, like how they choose the next Lama. It's really, really interesting. Children have to know where exactly some of the things are located and which of the objects was by the previous Lama or something like that. So leaving that behind, he talks about natal periods. So there are like 4 natal periods which determine our experience, because our mother is our world. So the first long part, the first stage is being, in the womb, in a safe, in safety, in, good experiences. But if there's some stress, the fetus is able to feel, and that's why the experience it has can be, coupled with different factors. So for example, like one of my friends had a germophobia. Like, she lived in New York, so I can explain that. But she was like overly, careful about being clean. Like, it wasn't too obsessive, but like it's like one of the spectrums of, obsessive compulsive disorder. And which is, like, sometimes if it makes your life difficult, it becomes a diagnosis. But many people live without diagnosis. So,
sorry, has it come after coronavirus, or?
Long before that. I've been there in 2018, yes, and she had that. So one time when I was listening to explanations of that theory about different natal periods, it had a little piece that was, I just remembered about her, because sometimes when a baby is in amniotic fluid and everything is generally fine, it feels okay. But sometimes there are stressful moments for a woman's body, sometimes expressed by a woman, so there's one dynamics, and sometimes suppressed, and that's why when you don't speak but there's a lot of stress, it's a little bit of a different combination of hormones. So sometimes if there's cortisol in your blood and you're nervous and you're stressed and you have that in your bloodstream, at that moment when a baby, if there's coincidence that the baby opens the mouth and breathes in, for example, amniotic fluid that has higher level of stress, I have to go now. Okay, I'm gonna be short with this then. So it means that like there's an imprint of memory that a fetus remembers as a feeling of being attacked by something that has been friendly this whole time. So like it's like a certain combination of factors, and that might cause some of the specific traits of a person later in life, including germophobia. Like, I'm in the world, I generally trust it, but I have a feeling that there's something wrong that might happen. But it's not something. It's subtle. It's not something crazy. So yeah, Ron had to go, so yeah, he really had to go. So yeah, I have to go now. Keep in touch, and thanks for your attention. Yes, thanks Ron for reading this lecture. If you watch the recording later, for everybody from Humanism Society, thanks for being with us and making so far to the end of the lecture and my long comments. And thanks everybody else. I think Islam, Mylin, and Yelena, you're still here. So if you want to chat, we can chat. If not, I'm gonna let me stop the recording first, and then we can have chit chat after party.